afternoon. Hello. I am very excited to see such a big group here. I'm really happy that you could come here, and I know that many have traveled from very far away. So it's a really great pleasure to have you here. So my name is Olga Vitek. I'm one of the co-organizers of the May Institute. So as I was telling some of you just now, it's the first time we're doing it on such a large scale. So it's a bit we're a bit nervous, but we're also excited. And so the goal of this 2.5 days in this classroom is to talk about statistical methods and R at the very beginner, very introductory level. And so we have several instructors. Lauren is one of them. Lauren, could you say hello? And we have several. So Dan, who is here, can you wave your hand? So she will be helping us with the R session, and several other people who will be coming in and out. So we'll introduce them as they come. And so for the first hour and a half, I will just talk about the introduction to what we're actually trying to achieve. Before that, can you hear me okay? Do I need the microphone? It's okay? It's fine? Okay, so why statistics? So the first question is, well, why do we actually need to spend 2.5 days and even more on that, right? And so oftentimes when I speak with my collaborators, they tell us that essentially we only need statistics when we have bad data, which of course I strongly disagree with. And so the argument goes that if our experiments are perfect and our measurement system is perfect and everything is well calibrated, then essentially the data will speak by itself and we do not need statistics. And my hope is to convince you otherwise. And so the reason for that is that variation and uncertainty are unavoidable. Just before I do that, so we have the slides on the GitHub, by the way. So you know how to access that, right?
I'll show the slide.
Oh, and you will share the slides, okay. So yeah, so you can have all the slides, of course, for you. So the variation and uncertainty are unavoidable, and there are all kinds of variation that we have to deal with. So of course there is technical variation, which comes from the fact that we have to handle the samples, store them, process the biological samples in ways which can affect protein abundance. There is also variation in instruments and different instruments themselves, but also the same instrument at different time points can vary in terms of elution time, how peak intensities, the strength of the signal and peak intensities, overlap of the signal and things like that. And once the data are acquired, so the next step is to analyze the raw mass spectra with computational tools. So find the peaks, quantify them, identify, and so on. And there is a wide variety of methods which exist for that. And each method and each tool makes its own assumptions. And these assumptions affect how the data are handled. And they affect the resulting quantities and our interpretation. So all of this is very important. But there is, of course, a lot of work now being done to improve the instrumentation, improve the experimental workflows, improve the computational tools. So all of this is happening. But I would like to argue that even if everything is really, done to perfection, we still need statistics. And the main reason for that is that our scientific question involves biological systems. So we're studying living organisms, and there is a natural variation between the abundances of protein from one person to another at any given time point, but also in the same person from one time point to another. And so even if our measurement technology is perfect, we still need to distinguish systematic signals, which are due to conditions, stresses, stimuli that we apply to the samples, from just random and natural biological variation that occurs in these biological systems. So essentially the point is that no matter what we do, we would need statistical methods to deal with that. And so that's why we're here. And statisticians, the goal of statistical insight into this is to ensure effective and reproducible research. Reproducible means that we get results which we can repeat once again, and effective meaning that we use our resources in the appropriate way. And so the statisticians contribute to this at least in 3 ways. And my hope is that during this 2.5 days or more, if you stay longer with us, we will go over all of these aspects in different ways. And so the very first place where statisticians contribute is the experimental design. And our goal is to design experiments which are produce results which answer the question that was asked, as opposed to having some other effects on the quantitative values which was not intended by the purpose of the design. And also efficient, so that we use our experimental resources and money in a way which maximizes the information content in the data. So this is where statisticians can really help. And if things get technical, and if it's too complicated and it's too much whatever math or too much information. So if there is one message that I really want you to take home from all of that, is talk to a statistician before collecting the data. So that's really very important. So we will hopefully see this in detail. The second thing is data analysis. So once we have the data collected, so we want to make sure that we claim changes, or we claim associations, or we claim biomarkers, or whatever we want to do in a way which does not depend on the person who looks at the data. And so statistical methods, you can kind of think of them as a body of methods for making wise decisions in the face of uncertainty. So we have uncertainty in interpretation, random variation. How can we make decisions which do not depend on the opinion of the individual person? And we all have opinions, right? We have our favorite proteins, we have our expectations. So how can we make sure that our conclusions are not too much influenced by that? And the last thing, which is also extremely important, is statistical tools, because it's one thing to have the data, it's another thing to have the method to analyze the data, but yet another thing is to have the tools which we can actually use in a way which is kind of streamlined and which does not depend, again, on the individual analyst. So statisticians contribute to this field also by developing tools which we can use. And so in my group in particular, this is one of our objectives, is not only to develop methods, but also develop software which can be available to everybody, open source and for free. And so this is really the motivation for that. And just to emphasize this point once again, that biological variation is really important. Every time there's a new measurement technology, every time there is new instrument, and everybody gets very excited and say, okay, we have new technology which will help us answer new questions and so on. Somehow people think that fundamental statistical principles of experimental design and analysis do not apply, but they very much apply. And so just as an illustration of that, so this was in 2011, just around the time when RNA-seq experiments appeared. And so everybody was very enthusiastic about the fact that we can quantify a lot more transcripts on a large scale with high accuracy. And as the result, we only need one replicate, which was a violation of the fundamental principles of statistical experimental design, to the point that this group of researchers felt compelled to write an editorial in Nature Biotech saying that sequencing technology does not eliminate biological variation, which turned out was a necessary message. And so that's what we would like to talk about. So for the hour and a half that we have today, what I would like to do is three things. So first, before we even start talking about analysis of data and methods, in math and R, and all of that, the very first thing we need to do is to translate our scientific question in a statistical question, because different questions require different statistical methods. And this is the very important thing we need to know. And then we'll talk about the experimental design. And at the end, I will talk about a case study which we were involved in recently, which kind of illustrates the need for the type of conversation that we have today. And I very much hope that you will interrupt me, because the point is not for me to talk at you, but to actually have a conversation. So please speak up at any time. All right. So the first thing is, so how can we translate the scientific question into statistical question? So when a collaborator comes to me, they don't come speaking in terms of, okay, let's say I have a normal distribution, I want to know something, right? They say, I want to find a biomarker of disease, right? Or I want to know if my knockout affected this pathway, So that's the type of way the question is framed. And so what is going on in my mind is I'm at least trying to classify the scientific question in at least three broad categories, which all have something to do with the class of samples or class of analytes that we're interested in. And so there are three broad questions, kind of broad statistical goals, and the first goal is class discovery. So class discovery essentially says, Well, I have some profiles. I have some samples for which I quantify some proteins. And by the way, for the sake of the argument, let's assume that for these 2.5 days, we do label-free quantification, so no complicated designs for most parts. So we'll talk about complicated designs later. So I have some profiles. So I would have, for example, a picture like that, where each row is a protein, each column is a biological replicate, and the strength of the color tells us the abundance of this protein in the sample, right? So darker blue means higher, lower abundance, darker red means higher abundance for this protein. And so the question would be, well, I want to know, for example, what are the proteins which have similar profiles in the space of the samples? And if proteins behave the same way across all the samples, well, maybe they're all involved in the same biological process. Maybe it's a process that's relevant to whatever conditions were here. Or maybe I'm interested in knowing what are the biological samples similar to each other in the space of proteins. And this is important, so maybe they're all cancer subjects, but maybe there's a cancer subtype. So knowing kind of individual subgroups of subjects which are similar can be useful. But the important thing is that it's called a unsupervised method, in that we say find patterns and find similarity. And there is no guidance in terms of who comes from which subtype of the cells, or which proteins come from which subgroup of proteins. So essentially we tell the method, go and find something, right? And this something may or may not be useful for our question. And that we don't know, right? But we say, okay, let's just find some patterns. So class discovery, and so this would be another example. So these are the profiles of protein abundances in time. By the way, this is taken from the manuscript by Niels Gellenborg, who will be speaking later in our main institute, I think early next week, or principal component analysis. So you are familiar with that as well, most likely. So each axis here is a linear combination of all the proteins. Each dot is a sample, and colors correspond maybe to the disease groups, right? And so we say, okay, if the colors are well separated, it means that our data are informative of these conditions, right? However, there's no known labels. So all variation, useful variation, random variation is treated equally. There's no concept such as error rate, right? Or how often I make mistakes, because we don't know what to compare it to, because we have no groups. And so this is typically useful for things such as data visualization, quality control, or representation of the data. But this is often not enough for most questions that we're interested in this field. So this is class discovery. So we look for things which are similar, and if they're similar, we say that they come from the same class. What this class is, we don't know. So the second statistical goal is class comparison. And so this is now a supervised method, in that we actually tell our method, these subjects come from healthy group, and this subject come from disease group. And again, just to be simple, let me talk about kind of clinical experiment where we look for healthy versus disease subjects, but you can kind of imagine that the same thing holds if you have knockouts or any other stimuli. It doesn't matter, right? So we have known class labels for each group, and so now our goal would be to look at abundances. Let's say here each dot is the abundance of the protein among all the healthy subjects. Here each dot is the abundance of the protein among each disease subject. And our question is, well, is the mean of the healthy systematically different from the mean of the disease? Or is the difference that we observe is consistent with random chance? Can it actually be an artifact of random chance? And so the methods that you would use for that would be something like t-test, analysis of variance, kind of methods like that. And the metrics that you would report would be p-value or a posterior probability. So these are very useful things. And you want to know if the change on average among the replicates was more systematic than as expected by random chance. And I would think that this type of methods are most frequently used in quantitative proteomic experiments. And so if you are doing things like initial screening, for example, you want to know which things are regulated by the stress, so this will be a good way to go. However, oftentimes people come to me saying, I'm looking for a biomarker of disease. And so there will be some analysis which produces some p-value, and then they say, we have proteins with small p-values, therefore I found a biomarker of disease. And I would like to argue strongly that this is incorrect. So proteins with small p-values are not biomarkers. And so let me give you an example, and for that, what we actually need to do is to think about what is a biomarker. And so a biomarker is something which is supposed to be actionable, meaning that a clinician should be able to make a decision for an individual subject. So what we see in a method such as t-test is that we compare the abundances of the protein on average between the subjects. But the clinician doesn't have the average subject. The clinician has a particular person sitting in front of them. And so even if on average there is a change, it may or may not be helpful for this particular person. And so the example is here. So for example, in this case, so we will say that the random variation is fairly small, so this change is unlikely to be an artifact. So if the person has this protein abundance, well, it's likely that the person is from this group, right, as opposed to from this group. But now if we're in this situation, same change in protein abundance between the groups, but now the person comes in with this protein abundance, well, it can be either this group and this group. So because we have many replicates, it's likely not an artifact that we have the change, but it is not actionable for the individual patient. So no understanding kind of average changes is not enough to claim that this is a biomarker. And that's what I mean by saying that different goals require different statistical methods, because a t-test will be fine if you just want to know what's regulated overall, but it's not enough when you want to actually use this in clinical diagnostics tools. So, and maybe one important thing, what I will be doing a lot here, talking about the difference between the means, right? And so you, of course, know, and if you were here just in the beginning of this week, we talk a lot about ratios, right? And so oftentimes in mass spectrometry, we actually talk about ratios, and I'm talking a lot about differences, so what's going on. So in statistical methodology, differences are much easier to deal with than ratios, just for technical reasons. And so, and the data that come from mass spectrometry have properties such that essentially it's much easier to work with this data on the log scale as opposed to on the original scale. And the log of the ratio is the same as difference of the log values, right? So you remember this, right? So if I have well, so I'll just tell you like this, right? So log A divided by B, it's log A minus log B, right? So on the log scale, looking at the differences is the same as looking at the log ratio. So when I talk about the difference here, think log ratio, right? It's the same thing. Okay, so this is an example where we have things which are differentially abundant but not predictive. But this would be the opposite situation where the things are not differentially abundant but predictive. So here's an example. So we have two proteins. So let's say blue dots are healthy subjects, red dots are the disease subjects. And so clearly, small abundances of protein 1 and protein 2 are associated with healthy, right? And large values are associated with the disease. So knowing the abundance of the protein is predictive of the disease. But here is the opposite situation. So you see that protein 1 spans the full range of abundances for both healthy and disease groups. And the same thing for protein 2. It spans the full range of abundance between healthy and disease. So neither protein 1 nor protein 2 will be differentially abundant. They will not have small p-values here because the means are very similar. However, as a multivariate panel of two proteins, they're perfectly predictive. So clearly, both small values and both large values are predictive of healthy, and the other of the disease. So of course, this is a cartoon. Real life is more complicated, but you see the idea, right? That not everything which is differentially abundant is predictive, and not everything that is predictive is differentially abundant. And so different tools are needed depending on our question. And so the statistical goal number 3 now would attempt to do that. So this is again a situation with two proteins. So let's say we have again healthy and disease. And so essentially here we ignore protein 1 and we only use protein 2 to classify healthy versus disease. And we're doing okay, right? But we have some red dots clearly in the wrong place, right? So we can do better. We can have some kind of more flexible function which uses both proteins to classify blue from red. And it's okay, and well, still some red dots in the wrong place. But now we can do something very, very complicated, which would really describe our points in great detail, and we will have no misclassified points. But this particular boundary is likely too specific to our data set, And it's likely that if we collect new data, it will probably not do as good job as on this data set. And so, and type of methods that you would use for this problem is more in the machine learning space. And so the goal of machine learning tools would be to find this boundary, which is complex enough but not too complex to do a good job. And so the type of metrics that you would see from that will be misclassification rates, sensitivity, specificity, and so on, which do not come with the hypothesis testing that we had before. And so if you're familiar with the fit-for-purpose kind of approach in quantitative mass spectrometry, where we use different types of experiments depending on the purpose of the experiment. So tier 1 and tier 2 would be the experiments which really aim at clinical diagnostics. So these are the methods which are required for these experiments. But if you want to do initial screening, like in the tier 3 biomarkers, where we only want to kind of eliminate, maybe doing some global shotgun experiments and eliminate, or rather focus on a subset of candidates which are promising, which we will target maybe more specifically in subsequent experiments with a larger sample size. So this hypothesis testing is appropriate. So it really depends on where we are in these experiments. So what I will do for today, I will actually focus on this middle ground on hypothesis testing. And in fact, for the rest of the 2.5 days, I think that's what we will be doing primarily. So just to finish that, so in terms of how statistical methods are used in the workflow, so no matter what we do, goal 1, 2, or 3, experimental design is key. And this is really going to affect our ability to make quality conclusions from this data. We always have to do quality control normalization no matter what we do. And then usually there is some statistical methodology and modeling involved. And so for class comparison, we would do hypothesis testing, adjustment for multiple testing, and this potentially informs the design of the follow-up experiments. For class prediction, what we will do, we'll still use statistical models to quantify the abundance of proteins so that we have one number per protein per sample, which is not trivial because in mass spectrometry, each protein has multiple features, right? And we may have multiple technical replicas and fractions and whatnot, right? So we need to combine that. And then develop classifier and use performance evaluation. And so when you talk to your computational statistical collaborators, if you can translate your problem in this language, it will really be very helpful in terms of communication and connecting what you need to what they know, right? So this will be quite helpful. Okay, so how are we doing? Questions?
Yes. So when you normalize, there are many ways to normalize.
Oh, yeah.
One of the proposals is, can you do normalizations in a series one after the other by different methods? And do you tend to over-normalize and then you get rid of all variations and all the class looks similar?
Yes. So actually normalization is just another instance of modeling and statistical inference, which is somewhat underappreciated sometimes. And just like any other statistical model like we had here, right? So this is overfitting, right? So when we just adapt to our data too much, right? And it's not going to be reproduced. Normalization can be exactly like that. So, and I think we will not have a chance to see it here, but we'll have a chance to see it in the next 2.5 days. We will talk extensively about normalization. And in fact, we are very excited that Rafael Irizarry, who is an expert in normalization, will give a keynote lecture on this topic.
I have a question about the imputation. Sometimes I'm very struggling when you imputation some data, means imputation my missing value. It actually scrub everything. So is it good for actually doing imputation or not?
So, well, again, this is something that we will talk about next week. But the short answer is that anything you do to your data implies some assumptions and some statistical model. And if this is not explicitly stated, it doesn't mean that the assumptions are not made. So sometimes people make assumptions without actually realizing that. And so your choice of handling missing values is essentially means a choice of assumptions you make about the data. And of course, what you assume about the data can have impact on your conclusions, right? And just imagine a situation when you have a lot of missing data, so it means that you make a lot of assumptions, and at some point your conclusions reflect your assumptions much more than the data, so you don't need the data, right? So you don't want to be in this situation, so there are some things that you can check before imputation to see if this is reasonable for you or not. But again, this is a topic for the next 2.5 days. So
size of data, maybe small size of data, you can actually check, but you may be handling maybe.
For sure. So that's a problem, but at the same time, you know things about your experiment, right? So you can kind of imagine. So the biggest thing is to distinguish missing completely at random, missing not at random, and then missing because of limits of detection. So missing completely at random can be in situations where you have peak overlap and you cannot reliably quantify the individual peaks, right? Missing below limit of detection and how you deal with it will have a lot of implications for the analysis. Next week, we will spend a full day on that. Sorry. Maybe we can give you a crash course during the break or something like that. So these 2.5 days, we will talk really about the basics of the statistical methods, because the fact is that these are not simple methods for missing values. And so we need to build the background, right, for the first things. Okay, so this is, but so the focus of our 2.5 days here is make sure we get going on R, and make sure we get kind of full understanding of like hypothesis testing, like things which sometimes people think we know what we're talking about, but then we realize that we actually have different ideas about them. Okay, so the purpose of this part here is talking about the experimental design. So before any data, right? And so again, what I would like to do is to tell you what's going on in my mind when a collaborator comes to me and says, I have some healthy and disease subjects and I want to design a proteomic experiment. So what is happening in my mind is this. So let's assume just we have two groups, right? Healthy and disease. And so I'm imagining that there are two populations of individuals out there which are identical for all practical purposes which can affect the abundance of the protein, such as age, gender, other diseases, lifestyle, whatever you can think about, except for the fact that this population is healthy and this population has the disease. And so we want to know if the proteins that we quantify change in abundance between these two populations. Now, these populations are very large, so we cannot really measure every single person in this population. So what do? We include some of the subjects from these populations in our study. And so because we include randomly, and because there's a natural variation of protein abundance from one person to another, if we want to repeat this experiment, we will have some other people in the experiment, so we'll not have the same protein abundances in our subjects. So this is the first layer of variation. This is the biological variation. But this is not the full story because we still don't know what the protein abundances are. So we actually need to quantify them using a measurement technology, which for the moment is subject to noise and variation. And so this is the second layer of variation where we quantify the unknown abundances of the proteins using the technology which itself can have some noise. And so our goal now, and the important thing is that there may be some mean abundance of the protein in healthy and diseased subjects. We assume that there are some numbers, they exist out there, but we will never know what they are. Same thing in our subjects which we selected. There are some actual numbers of mean protein abundances in them, but we will never, ever, ever know what they are. What we do know is the data that we collect from our instruments. And so our goal is to take what we know and make the conclusions about what we're interested in. And so using the data, so the things which we see, and make conclusions about what we don't see is called statistical inference. And so our goal is to take the data and make conclusions about the unknowns. And so this process has two problems potentially. One is bias, and the other is inefficiency. So bias means that our conclusions here are systematically different from the truth. And this typically has something to do with experimental design. And so an example is a study that I personally was involved with, where we had subjects with diabetes and healthy controls, and so we did, all this analysis and so on. And only later we realized, because we didn't have these annotations, that most healthy subjects were Caucasian and most diseased subjects were African American or something like that. So this is an example where you have a systematic difference between the truth because there was some additional forces built into the study, right, beyond what we were interested in. And the bias is the worst possible thing because bias is reproducible. So if you now repeat the experiment using the same approach, you will see exactly the same difference and you will say, great, I could reproduce it, but it's systematically wrong. And so the goal of statistical experimental design is avoid these situations because if you do have bias in your data, no amount of statistical sophistication can correct So it is really important to make sure that upfront you protect yourself against this type of things.
So is the bias reduced? So this is a tier 3 type of experiment, right?
So now this is actually type 2. So it's class comparison. So we compare the average of these people to average of these people.
So if your samples are genetic samples, so as compared to like treatment, so usually you have treatment
Right.
treated. So is the bias reduced in genetic samples?
It doesn't matter what your condition is. What matters is how you selected your subjects from the population. So if the subjects came from the two populations which are identical in terms of other things which can affect protein abundance, just one has this locus versus this, right, or something like that, you're fine, right? So it doesn't matter what is the type of condition. It's more like what is the variation around these conditions, and you don't want this variation to be very different from this variation.
And if you have a single mutation, that has less bias, right? Rather than, for example, if it's a disease condition, then you might have
Hold that thought. So I will spend more time on that, and maybe this will clarify some of the things. But so one comment that I often get kind of here is, well, we don't have the possibility to select randomly subjects from the population. We have to work with clinical samples as they come, from the clinic, and so on. And so then what is sometimes can be done is to work backwards and say, okay, this is the type of subjects which I can access. So what would be the populations that the subjects can reasonably represent? And what would be the potential sources of biases? And essentially you have to work backwards and see what are the potential dangers associated with them. Just spilled some water here. Sorry. Okay. Too enthusiastic. Okay, so let me tell you now what is going on in my mind, but more kind of mathematically. And I promise you there won't be much math here, but just so you know how statisticians think so that you can talk to them more effectively. So what we actually do is, oh, thanks. Thank you. So what we assume is that there are some constants right in the population. So this is the mean abundance of the healthy, and this is the mean abundance of the disease. And just so you know, in statistical kind of convention, we use Greek letters for things we don't observe and Latin letters for things that we observe. So there are some mean abundances of proteins in two populations, which we don't know. Now we have some underlying abundances in individual subjects, which we're interested in. What we actually observe is maybe peak intensities from multiple peptides, multiple technical replicates of the same subject. And we have some kind of summaries which inform us of the difference between the two groups. And our goal is to use these summaries to make the conclusions about what we are interested in. And so the bias means that this is systematically different from this. And in efficiency, I forgot to mention this, it means that essentially our summaries are so noisy that we cannot say anything reliable with respect to the question of interest. And so the second goal of statistical experimental design is to allocate the resources to minimize this uncertainty. And so this is another place where statisticians can help, because another comment that I often get is, I don't like to work with statisticians because they ask for replicates. So it is true that we need replicates, and I hope to convince you, but at the same time, there are statistical techniques which can get you more information content from the same number of replicates if you understand the structure of variation in your experiment. And this is where statistical experimental design and kind of statistical insight can help. Okay, and so, but before we can go into that, so there's two problems. So bias and inefficiency can be prevented with three fundamental principles of experimental design. And I hope that you know about them. But bear with me because it's really an important conversation for our field. So the first principle is replication. And so I hope I don't need to convince you by now that we need replicates, but let's just kind of look at this once again. So let's say we have some simple experiments. So label-free, let's say we have one peptide per protein per person, right? So just one number, and I have one replicate. And so this is the log of abundance of the protein in the healthy group, and this is in the disease. is it systematic or is it an artifact of random chance? What do you think? Who knows? That's exactly the point. We do not know because it depends on how much random variation is associated with that. So in this case, exactly the same difference has very little random variation around this. So this is not likely to be an artifact, right? Here, exactly the same change is associated with a lot of variation. So it is more likely to be an artifact. So one role of replicas is allows us to actually distinguish what is systematic and what is an artifact, right? So this is very important. And the other role of replicates is that it minimizes inefficiencies, because if we have more replicates, we will be very, very confident in this mean, very, very confident in this mean. So it will be easier for us to claim that this is systematic if we have many replicates, even if there is a lot of variation here, right? So this is a very important part of replication. And so an important thing here is, remember I was just saying earlier, on the log scale, differences, they are log ratios, right? So using just the extent of difference alone is not informative. So in our language, it translates to using fold change cutoffs to claim regulation, right, or to claim impact of the whatever stress on protein abundance. So essentially what this means is using two fold change cutoff or any favorite fold change cutoff is not appropriate, because the same fold change cutoff may or may not be systematic depending on how much variation is associated with this particular protein. And we will see it very well later. Okay, so, and of course, because we have two layers of variation, remember biological variation and technical variation, we have two types of replicates. We can have biological replicates, so multiple subjects, and technical replicates, which are multiple, sample processing instances, mass spectrometry runs, and so on. And long story short, biological replicates are a lot more important. So if you have a fixed number of runs available, you will be much better off statistically to have each run dedicated to a different individual, as opposed to take one person and measure this person really, really, really well, but you still have only one person, so you don't have any biological replicates. So that, I hope, is clear. So the second fundamental principle of experimental design is randomization. And so randomization is, the main purpose for this is to prevent bias. And so here is the example. So let's say we have a healthy group, again, one number per protein per person, and all the healthy people were measured on day 1 and day 2. And then all the diseased people were quantified on day 3 and day 4. So what happens now is that this difference between healthy and disease can be due to two sources. One is to the status of the disease, but the other is to the time of data collection. And this would be an example where no amount of statistical methodology will correct for that. So you'll probably tell me, oh, we can normalize, right? But again, normalization makes assumptions. And it will probably make assumptions such as the effect of day is the same or similar for every protein in the sample. So very quickly you run into, assumptions which are really, questionable. And so we're much better off to remove this type of effects in the first place. And so a good way to do this is to essentially take each sample and randomly assign it to any day of data acquisition, right? So just essentially make the permutation of them. And we will have some ways to practice this in our later on. And so now we see that our samples in each group are spread out across different time of data collection. Now the kind of side effect of that is that now we have a lot more variability, because now not only we have the biological variation in each group, but we also have a day-to-day variation which contributes, right, to our measurement quality. But at least we don't have the bias. And so when we have two effects that affect the difference between the groups, it's called confounding, right? And so the randomization prevents us from this situation. And the good thing about randomization is that it can also help us avoid biases like that from sources that we're not even aware of. So day effect, it's kind of a well-known situation, because time of data collection is a proxy for very many things, right? And very many things can go wrong in time, right? But there may be some other things which we may not be even aware of. And so by randomly assigning samples to all of the steps of handling, storing, and collection, helps us avoid that. So, and again, because we have two layers of variation, we have two types of randomization. So first, we randomly select subjects from the population, and second, we allocate the samples randomly to all the processing steps. And I forgot to mention, this is the reference which talks about this quite extensively, which is getting old by now, but fundamental principles still apply, so it will be relevant, so I invite you to look at that. And so here's one example. So I really like this paper from statisticians in MD Anderson Center. It's very old, so it was with old technology. I think it was CELD, yeah, but it doesn't matter because still the same thing. So this is a group which does a lot of consulting work, so they have to do data analysis for their collaborators. And so one of the projects was where they had samples with 5 types of cancer, and it was blinded, so they didn't know which sample came from which type. So they did class discovery, right? So they looked at dendrograms. So, well, first of all, they did all the right analysis things. They normalized, picked, quantified, and all of that. And then they did class discovery, and they were very surprised to find 6 groups and not 5. And so they were very excited because they thought maybe they found a cancer subtype, which is quite interesting. But so what they did next was the same plot where the x-axis is the same samples as here, but the y-axis is the day when each sample was collected. And so it's very obvious that the main source for clustering is the day of sample collection. So my example here is kind of a cartoon, but it's motivated by that because day of experiment is a very strong kind of indicator of many things. And so I'm really grateful to them because they put together a manuscript which describes all the failed examples. So all the examples where things didn't work out, and we can learn from them. And so, well, this is one, but this is another one. So it's from a different area, but I also find that's extremely important. So this is taken from a blog by Jeff Lick, who is a very prominent statistician at Johns Hopkins. And so this was a manuscript published in 2007, where researchers claimed, so they did study of gene expression between Asian and European subjects, and they found that 78% of genes are differentially expressed. So 78% of genes are differentially expressed between Asians and European subjects. That's a lot of genes, right? And so, of course, they were quite kind of interested in that, and they wanted to reproduce the analysis and see what exactly is going on. And they did that, and they did found that there were big differences. Luckily, this particular manuscript made the original data available, and this was the time of Affymetrix microarrays. And in Affymetrix microarrays, the actual array in a corner has a timestamp. So in the array, they can read out the time of hybridization. And so what it turned out that the Asian subjects were profiled 1 year prior to the European subjects. So no wonder things would be different. Right? But the thing is that this manuscript didn't even report that, and didn't document it, and it took all the way going to the raw data to find it out. So it is really an important lesson from here. I don't know what happened to this paper. Okay, so this is, so any comments so far? Okay, fundamental principle number 3, and this is blocking. So this is the third part which requires more statistical expertise. So I would say that if you can collect replicates and randomize, you will be quite well off already. So this is like the basics. The goal of blocking is to produce further improvements. One is in eliminating bias, and the other is reducing inefficiency. And so here's the idea. So blocking is applied in situations where you have some systematic things which affect the abundance of proteins. You know they exist, they're known sources. It just, you cannot do anything about them. You cannot eliminate them by things like better, instrument tuning, or something like that. So here's an example. So remember, I was saying that if we have a day effect of data collection, and if we now randomly allocate samples to dates, we inflate our variability because now our variation has the person-to-person variation, but also the technical day-to-day variation. So one way to deal with that is actually impose a restriction on randomization. So blocking is essentially a restriction on randomization, where we say, okay, let us impose on each day that we always have one healthy and one disease subject profile. Now, I will take a random subject from the healthy cohort, a random subject from the disease cohort, but one from each. So now on the second day, I will do the same thing. Let me have one healthy, one disease. So I would rearrange randomly the order in which they're profiled, but we'll always the same number. Same thing for the third day and same day for the first. So it does two things. So if you have a very small number of replicates, even if you randomize, by random chance, you can still end up being like more on day one and less on day two, right? So it really imposes this restriction. You have exactly the equal number on each day. So it's a good thing. But the second thing you can do is now, because you imposed restriction on randomization, instead of comparing the mean of this group to the mean of this group, what you can actually do is to look at the differences within each group. And you see now that within each day, the difference always points in the same direction. And so it points to the fact that the disease group is always higher than the healthy group. And so if you compare the mean here versus mean here, you will have too much noise to see the signal. But if you now work with the differences, the signal will be much more clear. But this is something which requires both understanding of your experiment and understanding of how the data will be analyzed, right? Because here, even if we allocate the data systematically, if we don't analyze it correctly with the differences, we would not be able to see the signal. And so this is a cartoon, so we can have, so each day here is a block. So a block is a instance where we restrict randomization. It's easy for me to draw a picture with one sample from each group in a block, but generally you can have much larger blocks. So what if you can have 10 runs in one day? Well, you will have 5 runs from healthy, 5 runs from the disease, randomized. Next day, 5 other healthy, 5 other disease, randomized, right? And going on like that. And the same thing, so we'll be looking at differences essentially within each block and combining them. So that's what we do. And the same idea. So because we have 2 levels of variation, we have 2 types of blocking. So we can block with respect to biological replicates. So essentially looking at characteristics which we know affect protein abundance, and age and gender in human samples is like the hallmark, right? So age and gender affect everything. So usually in clinical literature, you want to look into that. And because we have technical variations, so technical replicates, also we can block with respect to technical replicates. So let me give you just some examples. But before that, so this is another study from the same paper, which is also telling. So same idea. So they had cell death, now 2 types of cancer, also blinded, did everything right, had 2 groups, were very happy. And then when they unblinded, they realized that this was the protocol change, which of course happens, right? So imagine, for example, that you run a very long-running experiment, right? So you are bound to have new batch of materials, right? Or new operator coming in, right? Or something like that. So, and we know that these things will affect the abundance, but we have to work with that. So a good design would be to have an equal number of healthy and disease before the protocol change and a good number, the same number after, and then combine essentially the differences. So this would be quite important. So now for matching specifically, and this is also from a review like a few years ago. It's a very brief review, but you can kind of look at that. So we have 2 populations, what I was kind of showing earlier, and we say they're roughly equal, right? But they're also very heterogeneous. So you were just saying earlier, right, that people have all kinds of, properties, right, and all kinds of things, including genetic variation, right? So if our sample size are very small, even if we randomly select things correctly, just by random chance, we can all end up with healthy, which are all male and, older, and then disease, which are all female and younger, right? And so so if we have a very large sample size, it will average out eventually. But small sample size, it's still a danger. And so blocking with respect to biological characteristics is called matching. And so what we can do, we can impose restriction on randomization, where we'll say, okay, I want to have young female from each disease group. And then I want to have an older female from each disease group. And then a younger male, and so on. And by the way, you don't have to have an equal number of each of those blocks. But the important thing is that each group has the same distribution, right? And so that allows us to both eliminate the bias, and if we can analyze separately each disease group, we can reduce variation. And so this is just one example from a old collaboration that we had. So the numbers are just for illustration. It was not exactly like that, but so it was a collaboration with the Munich Heart Center where clinicians were storing blood samples of people who come with various types of heart problems. And so they had essentially a repository with these disease groups and this number of samples available in the biobank. And of course, so age and gender are important. So our collaborators decided that we will use this age cutoff and this gender. So we have four groups for each combination of age and gender. And so one way to block with respect to these characteristics is essentially randomly pick samples such that the number of samples in each row is the same. And so here, just as an example, we have 3 samples from each disease group with these characteristics, and then 2 samples from this disease group with these characteristics, and so on, right? But so then if we compare this group to this group, they all have the same distribution with respect to age and gender. So if you ignore this during the analysis, it will avoid bias. If you have a large enough sample size and you actually account for these covariates in the analysis, you can also improve your efficiency. So it's a combination of experimental design, allocation of resources, and analyzing the data in a way which responds to the design. And so this is why it's really important to talk to a statistician, because if you don't know how the data can be analyzed or will be analyzed, it's really not easy to know how to allocate the resources to that. Comments?
So,
Yeah.
so in a large study, when there is a instrumental drift going on, so what does it qualify as, inefficiency or bias?
Bias, because it systematically affects the signal. And so essentially instrument drift, well, again, a proxy for instrumental drift is a day, right, or time of data collection. And so what we will do, and I think we will actually have quite a bit of hands-on exercises for that, is to use essentially time as a block. So time is continuous, so you may have some flexibility on how you set up the blocks, also depending on the plates that you use. But let's say you have 5 disease groups, like here. So what I would do is to have 1 subject from each disease group, randomize, next 5, 1 from each group, randomize, next 5 from each group, and randomize.
Yeah, so my question was for, okay, this is great because it's all for patients in vivo studies. For in vitro studies, for example, if you have a large experiment, you have many conditions, I guess if you want to repeat that, you want to do the same experiment, the same exact experiment on, let's say, 3 different days or with 3 different passages of the same cells,
Well,
right?
So when you work with cell lines, it's actually difficult. So if I go back to my very beginning here, so essentially you would need to ask yourself, are there populations of different growths of cell lines, or are they completely exactly identical, right? So are you working just comparing this, or are you working comparing this? And what we found was that growing different instances of cell lines contributes to understanding how much variation you have, as opposed to take the same cell line and just do more mass spec runs on them. So, but you're right that it's sometimes less obvious what constitutes new biological replicant in the population of cell lines, for sure. Oops. Uh-huh. Mm-hmm. Yeah, so sometimes, so you're saying, can we have more people in the control than in some other disease group, right? You can, provided that, again, you have the same distribution of age and gender in the control. So you can multiply each of these numbers by 10. It will still be the same distribution compared to that. So you can do that. But what you don't want now is to have control which are all young and female and disease which are all old and male. And so sometimes it actually requires some adjustments, right? And I actually had a collaboration like that where pretty much this happened. And they tried to kind of encourage us to say, okay, well, let's just do the analysis anyways, and we will do some, adjustments for covariates and all of that. And I said no, because I would rather have the data speak for itself than try and come up with some models which may or may not be really appropriate, especially when the sample size is not very large. And so we went back and we asked our collaborators to have more representatives of different disease groups, and it was much better. So if you can, and generally I find that it pays off a lot to design your experiment correctly to avoid having to justify some of the choices which may be somewhat arbitrary, than trying to kind of, save it somehow later. So this is really the key, right?
Like in some biomarker studies, the patient samples that you have like a bunch of diseased ones and not enough control, or maybe the opposite way.
That's fine. As I say, provided that, so it's not the number in each group, but it's the properties within each group that matter, right? So if you have more here than there, it's okay, but you don't want to have very different people here from that, right? And so what it may require you to do is to actually eliminate some of the subjects from the study if they create too much imbalance. Like, for example, if, just to give an example, if all your disease subjects are female and your controls are female and male, it may be a good idea just to eliminate males completely, right? And just focus your study on the females, and then your populations are defined as female subjects as opposed to any subject.
Sorry to ask another question. Have you encountered a case where you don't have any information about the samples? You only know whether they have the disease or not, but you don't know how old they are or what gender they are? I just have to do this.
Everything can happen, but you just have to manage your expectations with respect to the answer, right? So you can still proceed with the analysis and do things, right? But how much you expect to be informative and reproducible later on will depend on what's in there, right? And so personally, I advocate very strongly understanding exactly every single step which occurred before the analysis, because it can have a huge implications on everything. Yeah. Okay, let's make sure we stay on time. So just to finish with blocking, so there's just in terms of the language again. So I just mentioned that we have two types of blocking, right? On biological characteristics, so it's matching, but there's also on technical characteristics, right? And so we just talked about the day of data collection, but there's a very special instance of blocking which is used a lot in mass spectrometry, which is multiplexing, where we combine samples from different sources, right? We label them different and then we pool them, right? And so then we acquire multiple samples within the same run, and we look at the mass difference to see who came from what, right? And so multiplexing is actually another type of blocking. And what you can do is multiple things. So we can have, if we have, for example, using metabolic labeling, right, like SciLag, for example, right? So you can look at differences between two peaks within each run and combine that. So run is a block. That's exactly what we just talked about. And another example is, which is often used in the SRM experiments, where instead of having two endogenous samples mixed, you add standards. So, for example, isotope-labeled reference peptides spiked in constant concentration for each endogenous peptide. And so this would be an example of this type of blocking. So again, a run is a block here. And if you just compare the endogenous blue dots here to the endogenous red dots here, so comparing the blue to the red line, you won't see very much difference. But now we spiked the reference analytes in each sample in a constant concentration. So because we have so much difference between these dots, it means that our quantification has a lot of technical variation here. But again, remember, we often talk about ratios, right, of endogenous analytes to reference. So on the log scale, these are differences. And so what we're doing, we're comparing the difference, the deviation of each blue dot from its reference among healthy to the deviations of each red dot from its difference among the disease. And so what we see now here is that these deviations of the red dots are systematically larger than deviations of the blue dots. And again, it points to the fact that this protein is upregulated in disease compared to healthy. And so this is, again, a combination of experimental design where we actually spike standards in each mixture and analyzing the data appropriately to recognize that. And so I hope that probably early next week we'll have a chance to talk about that. And so I would like to kind of advertise here. So yeah, I would like to advertise here this series of vignettes, which there are a lot more than what I show here. So it's Nature Methods in the last couple of years started doing points of significance vignettes, where it's like 1 or 2 page summaries on different topics of statistics. And it's very nicely illustrated. And I'm trying to use some of the figures from these vignettes as much as possible so that you actually have a text that goes with that you can look at. And I'm very excited because Naomi Altman, who is one of the lead authors of the series, will be speaking in our May Institute next week as well. So we will have her talk about multiple testing and false discovery rate. So we will be referring to this as we can. And so also to kind of emphasize the point of the importance of experimental design, this is also from Jeff Leake and Roger Peng, pretty old by now, 2015. I hope you can see what they're talking about here is the whole process of, doing kind of experimental design and data analysis. So you start with the design, collect the data, raw data, data cleaning, tidying it, exploring statistical methods, statistical models, summary statistics, inference, and p-value. And what happens a lot, and it's my practice as well, that at the end of the day, you get the p-value that you don't like for some reason. Maybe your positive control is not significant, right? Or something that, nothing is significant, even worse, oh my God, right? So nothing is significant, right? And so what people do, they essentially blame p-values. When in fact, all of these things happened before, right? And problems could have occurred from the very beginning, including not enough sample size, including biases, right? And so this is really a very important thing to remember, right? That it's not all about just fixing p-values somehow, that there's really a lot of work happens here. Okay, so, we're good. We actually have some time. So, any questions? Yes?
Going back to replication, from statistician's point of view, usually with NGS experiments, you just have one replicate and then you do a validation. Is that?
Well, actually, so it depends. So let's just think about the context of this, right? And it really depends on why you are doing the experiment, right? So if this is your very final claim of evidence for whatever you're finding in this type of samples, and you are going to, I don't know, submit an FDA approval, right? Or you're going to write a Nature paper, right? Or something like that. Chances are that your differences are not systematic just because we don't know how much variation is around them, right? So it is certainly not enough. On the other hand, a lot of these studies, they're not just one shot, right? So you actually do it kind of in an iterative manner. So in proteomics, for example, what you would do is some kind of global discovery experiment, which may have a very small number of replicates. And you would look at it to say, okay, what would be the interesting proteins there? And you would narrow down your list of proteins to some small number. So essentially, these are hypotheses that these are some useful proteins somehow. And then you build your next experiment, for example, targeted experiment, where you will go now follow these particular proteins and, maybe with better SRM experiments, right, larger sample size, and you will probably narrow it down to small. So in this case, your initial screening experiment was essentially a hypothesis generation experiment, right? And your hypotheses, they can come from anywhere you want, right? Including the literature, right? Including some other technologies, right? If you want to do one replicate, do one replicate, but with the understanding that you may be wasting your money following up on something which is not really a systematic signal, or you are not detecting something in your screening experiment which could have been useful to follow up on. So at that point, it's just a judgment call, right? So what is a good use of your resources? So personally, I have nothing against these exploratory studies, because also you probably want to calibrate your instrument, make sure things work well, right? As long as it's not your final claim, that you have something systematic going on. Any other? Okay, so now we have a little bit of time left. So what I would like to do is to tell you about a study which in some way motivates the programs like this one here. So I was involved a couple of years ago, and several of my group members were involved in the IPRG. So it is a study organized by ABRF. So ABRF is the Association of Biomedical Resource Facilities. How many of you are familiar with ABRF? A few, the broad folks. So ABRF is an association which brings together primarily people who work in core facilities. So in industry or in academia. And so the aim of this organization is to establish the best practice. So they're not necessarily after, like cutting edge research and new kind of technologies, although it's also interesting, but the goal is, okay, how can we use the methods which exist now, which are fairly well established, and how can we use them in the best possible way, which is reproducible and accurate and so on. And so the organization created the studies. So IPRG, I think it's Informatics of Protein. I forgot what it stands for. But the idea is that these are groups which would come up either with biological samples or data sets where every member of the association gets to work with. And then they report their results, and we look at that and we say, okay, who did well, who didn't do so well, what's a good practice? And it's not a competition, there is no winner or loser, just a way to say, okay, probably sometimes there are multiple ways to work with the data and they're all good. Some practices are clearly not very good, and just bring up the quality of the work generally. And so the particular group I was involved with focuses on data analysis. So there's no experimental component, it's really working on working with the data. And so and the focus was to look for differentially abundant proteins. So if we ask people to work with data which detect differentially abundant proteins, we need to know how to evaluate that. So we need to work with a situation where we know the answer. And a good way to do this is to create controlled mixtures where we spike some analytes in concentration that we know. And so if we look for differences, we will know what is changing and what is not changing. And so in this particular case, we had a yeast background, and these proteins were spiked in yeast background, so in these 4 different concentrations. This particular pattern here, so where we have each protein is only present once in each concentration and across samples, and within sample, each protein has different concentration, is called the Latin square design. And it's actually a very effective design because you can look at many comparisons over many proteins in a very small number of groups. And then we have these 2 additional proteins spiked. So in retrospect, we found that the true concentrations were a little bit too high. Some of these changes were a bit too obvious, but still it kind of allowed us to have the right conclusion. So this is published, so the work was led by Mina, who is sitting right there. Mina, could you wave your hand? So the goal was to collect the data and provide the resulting raw data or some partly processed data to the participants and see how well everybody does at detecting changes. So that was the idea. And so just very briefly for those of you who are kind of more on the experimental side, so essentially on thermo instrument, Orbitrap, shotgun, important part, shotgun label free, right? So no targeted, so regular, no fractionation, nothing like that. So these experimental details, right? Okay, and then what we did also, we did some processing of the spectra. And so 2 things. So one was we had a identification of MSMS spectra so that we can identify the peptides. And so it was done with this several types of database search. And at the same time, we also looked at MS1 quantification with Skyline. So Skyline takes this database search as input, and then we found that many proteins within Skyline. And so the participants had a choice. They could either get the very raw data without any of this, or they can do only look with the results of the database search, or they could actually have MS1 intensities as input. They could start wherever they wanted. We didn't tell them, of course, which proteins were spiked and what the changes are, and their task was to see, okay, what is changing between conditions. So just kind of for fun, so turns out many people were from Europe for some reason. The majority were from core facilities, but also people doing research, or core and research. Many were bioinformaticians, but also some experimental people. Many had more than 10 years of experience, but second highest was 1 to 2 years of experience. So it was kind of fairly representative of the community. So this is the summary of submissions. So this is the first thing. So here, the x-axis is the participant ID. So this is blinded, so we did not know who is who. So we had like a person who would anonymize all the submissions. And the participants had to report the proteins they had, changes in abundance, and also they had to fill a questionnaire kind of detailing what they did exactly. So the first thing is just the number of proteins that they reported here. So this panel is people who went with intensity-based quantification. These people went with spectral counting, and this is some in-house hybrid approaches and not reported. So you see that the number of proteins varied quite a bit between the participants. And you see that the colors are kind of mixed. So these are the people which used our MS1 quantification. So their number of proteins is as provided, right? But others had a little bit more or a little bit less. So these people misunderstood the task. They only reported differentially abundant proteins. But it's okay. So you see already that just by looking at that, the number of reported proteins varies quite a bit between the submissions. Okay, and these folks found like a lot more proteins than everybody else using spectral counting. Okay, so the next thing, and this is probably the most important result, is we asked them to report the number of differentially abundant proteins among all pairs of comparisons. So it's choose 2 possible comparisons, right? And so here, the horizontal line is the true changes. So we know what is changing. So this is the number of true changes. And above 0 is the number of false positives, right? And you can see how much the results vary between people who do this type of analysis a lot, right? And so, well, sometimes people missed some of the true positives, but it's okay. But you see, like, this group reported 2,000 differentially abundant proteins, right, out of the 4, 5, 6 that we spiked, right? And you also see that the colors are mixed, so it doesn't matter where they started from, right? So you could do well or you could do not so well, regardless of your starting point, right? And spectral counting or intensity-based or something else, right? That's why we do these programs. Okay, this was also interesting. So the next thing we asked them to do is to report estimates of fold changes. And so here we look specifically at estimates of log 2 fold changes, so absolute log 2 fold changes, but only among the background proteins. So, so they're expected to be small, right? Because it's background proteins. But you see that every single study had some fold changes which were large and other fold changes which were small. So there's quite a bit of variation for all of them. But people who did well, so the order is the same on both plots. So people who did well, they were not misled by these large variations in background proteins. But people who didn't do as well, they probably were misled by some of this variation among background proteins. So this is kind of speaks to this fold change cutoff, for example, right? That it can really be quite misleading. So we really need to do more than just look for changes between abundance. So that's the situation, right? And so this was probably the most kind of interesting thing for us because, of course, the next thing that everybody said, okay, so who did better? So which software tool did better? Was MaxQuant better than Skyline, you know? Or like, you know, so that was a very kind of typical question. So what we tried to do, and so this was work by Mina, we tried to parse the questionnaires to try to have some kind of system of what different people did, what they did to come up with these differentially abundant proteins. So I'm not sure you can read. So there's some things such as type of input, how the identification of spectra was done, how quantification was done, summarization, statistical inference, software inference, multiple testing, and so on. And we tried to kind of characterize them by some broad classes with the colors. It was actually very difficult for several reasons. One would be some people would just write in-house software, you know, R scripts. You know, it's really not helping, right? Because we don't know exactly what was done. Some people, we could just tell that sometimes they didn't know exactly what the software was doing that they used. So you could tell that the information was essentially not very complete. But the point that I think this figure tells primarily is that there are many colors.